Hello, my name is Evan Sukup, and I will be talking to you all today about hedgehog signaling. I will be specifically focusing on its key role in gastrointestinal development. Hedgehog is a group of morphogenetic proteins that regulate patterning through signaling pathways. There are 3 known mammalian hedgehog genes: Sonic hedgehog, named after the popular video game character; Indian hedgehog, named after a hedgehog species from Pakistan; and desert hedgehog, named after a hedgehog species located in Egypt. Sonic hedgehog and Indian hedgehog are very important in development by playing a key role in endodermic signaling, which patterns the developing gut tube. Due to its key role in development, mutations in sonic hedgehog and Indian hedgehog can lead to severe malformation of the gastrointestinal tract. Similarly, hedgehog signaling regulates homeostasis in the stomach and intestine of adults. These first 2 images I'm about to show present a simplified version of hedgehog signaling. There are 2 transmembrane receptors important in hedgehog signaling. First is patched, abbreviated PTC, and depicted in red. Patched is a 12-span transmembrane receptor with 2 extracellular loops that control hedgehog binding. Second is smoothened, abbreviated SMO, and depicted in blue. Smoothened actually transmits the hedgehog signal. Smoothened is not where hedgehog binds, but hedgehog does control the activity of smoothened. Without the presence of hedgehog, patched inhibits the signaling of smoothened. Patched does this by pumping inhibitory molecules across the cell membrane where it acts to inhibit smoothened directly. Upon hedgehog, depicted in yellow here, binding to patched, patched function of inhibiting smoothened is blocked, which allows smoothened to activate and transmit its signal function. Now these next 2 images show hedgehog signaling in a bit more depth. As we can see in this picture, hedgehog is not bound to patched. While hedgehog is not present, smoothened locates itself mainly in the cell membrane and intracellular vesicles. Gli2 and Gli3 are phosphorylated by protein kinase A, abbreviated PKA, and then processed by the proteasome. Gli2 is fully degraded, but Gli3 is only partially degraded. The result of this partial degradation is that Gli3 protein lacks a transcriptional activation domain. This represses the expression of hedgehog-targeted genes. In this next image, we see that hedgehog is now bound to patched. With hedgehog now being bound to patched, patched no longer is repressing smoothened. With smoothened not being repressed, it moves to the primary cilium where it can activate the Gli proteins. Now that the Gli proteins are activated, transcriptional activation is now possible and proceeds. Gli2 and Gli3 are key in transducing the hedgehog signaling in the gut. Now with both of these transcription factors active, cell proliferation can proceed and new cells can be produced, whether that be in a developing embryo forming its gastrointestinal tract or an adult maintaining homeostasis of their gut. Next we see here the abbreviation key and the works cited page. In conclusion, I hope I was able to teach you also about the importance of the hedgehog signaling pathway and its key function in gastrointestinal tract. Thank you all so much for your time.